So, welcome back, everyone. And today we'll be talking more about, we'll be going deeper into the explanation of plastics and degradation. Like I earlier mentioned last week, I talked about, sorry, I said last week. As you earlier mentioned, in the last video, I talked about how plastics are accumulated in the environment and how they're detrimental to human health and the ecosystem at large. So this slide is showing us different conventional methods and their effects and how effective they are. Different conventional method, their effects, and how effective they are. Now, Bara et al. In 2018 made us understand that the accumulation of plastic waste is an ever-increasing, pervasive, anthropogenic threat for survival of natural ecosystems due to heavy usage of plastics. And then, now, so plastics actually are non-new, but the increase in usage is dated back to COVID-19 era. During the era of COVID-19, the demand for plastic increased, like I earlier mentioned. Now, there's a study in New York in 2021 where the University of New York, a pediatrician in the University of New York, confirmed that plastics were found in the fecal samples of a newborn child. You can see how detrimental plastic can be. Now let's go deeper into the explanation of this figure. It's showing the effect of plastic emission in the environment. Now let's pick it from left to right. Now, so it begins by contamination. So what does plastic do? So this figure is just more of an explanation of the effect of plastics. Now, the other time when I talked about when we dump them at the dump site, like when we leave them in the dump site. So now this figure is divided into three parts. This part is the effect of dropping them at the dump site, just dropping them. This part is the effect of recycling them. What happens when we recycle them? And then this part is talking about what happens when we burn them. You can see incineration. When we burn them, what happens? Now, when we just drop them on the soil or in water, now they cause contamination of groundwater. They also affect soil structure in turn. They also affect soil microbial diversity. And now these plastic accumulate in food chain and thereby they're being ingested by marine organisms, like by aquatic organisms like fishes, and then they eventually affect marine life. Also, now this is effect of throwing them just on the soil. It also comes at this stop and say, oh, what happens when we put them in landfill? Now, some of them affect when they are discarded. From the landfill, when we have rainfall or anything, some of them that are dropped in the landfill can go into the ocean and affect aquatic life. Now we're like, okay, let's leave okay, now we agree that we cannot put them in the upside. What do we do? Some school of thought are saying that, okay, let's recycle that. But of these 8.8 million tons of plastic every year, how many can be recycled? With a new product, closed loop recycling, most of them, only 14% of them. Only 14% of them can be recycled. This is an example showing that 6.3 billion tons of plastic waste are tried at, pardon me, 6.3 billion tons of plastic waste are being recycled. Now, only 14% of them are effectively recycled. Good. Now, okay, this figure helps us establish that recycling is not effective because of 8.3 billion, only 40% are being recycled. Now we're like, okay, let's burn them and then incineration. Burning causes acidification of soil and water. An effect. A contaminated soil by heavy metals. When we burn them, heavy metals contaminate the soil. And again, it eventually causes global warming. So just like I explained, this is a pictorial figure explaining what I said in the last video, why we need to look for, why it is important to look for a safer method. So that because these conventional methods are not effective, that is what brought us to the idea of using microorganisms. And then the use of microorganisms can simply be referred to as biodegradation, biological degradation. Biological here are the microorganisms, and degradation is the breaking down of plastic. Now, biological degradation of plastics involve excretion of extracellular enzymes by the microorganisms, attachment of enzymes to the surface of plastics, hydrolysis to short polymer intermediates, which are ultimately assimilated by microorganism cells as carbon sources to release carbon dioxide. Now, in the last video, I mentioned how microorganisms are economically friendly and financially friendly. They do not cost a thing, and then they do not affect the environment. This is why it is important. This is why it is important to use microorganisms. This is the reason we are focusing on microorganisms, because they do not cause a thing and neither do they affect the environment or humans. Now, plastic degradation is a multiple step biological process in which microorganism uses plastic polymer as carbon and energy source. You know the importance of carbon and energy in the metabolism of microorganisms. So now, these microorganisms use plastics as carbon and energy source. However, because plastics such as polyethylene are chemically stable, microorganisms cannot directly take them up into their cells. Instead, degradation occurs through a series of extracellular and intracellular processes. So these are various extracellular and intracellular processes. Now let's take them step by step, one after the other. The step one, which is biodeterioration. This involves surface colonization and initial damage. This process begins when the plastic surface is exposed to abiotic environmental factors such as ultraviolet light, temperature, oxygen, and photooxidation. Now, these factors, that is the oxygen, temperature, ultraviolet light, they weaken the polymer by introducing oxidized functional group and creating small cracks and pits on the surface. Although these cracks or pits do not completely degrade the plastic, it makes the material more susceptible to the microbial attack. That is, it makes microorganisms able to attack the plastic. Now, because this environmental factor already make the plastic surface more susceptible, microorganisms then attack to the weathered surface and form a biofilm, allowing them to remain in close contact with the polymer. Now, this biofilm that they form allow them to remain in close contact with the polymer. The attached bacteria secrete extracellular depolymerizing enzymes. You know now that first, the microorganism abiotic factor affect the surface of the plastic and make them susceptible. Now, the microorganisms are able to attack the surface. When the microorganism attacks the plastic, they create biofilms. Now, they now secrete this microorganism, either fungi or bacteria. They now secrete extracellular depolymerizing, enzymes, such as the oxidases, the lactases, the monoxygenases, the oxygenases, esterases, lipases, cutinases, amongst others, depending on the polymer type. Now, depending on the type of plastic, is the type of enzymes produced, the type of enzymes released by the microorganism is dependent on the type of plastic that they're attacking. Now, these enzymes that the microorganism release, they begin to attack the polymer surface, initiating deterioration and initiating reduction, initiating degradation. So now at this stage, the plastic is not yet, now this is the polymer here, this bacteria attacking it. Now, at this stage, the plastic is not yet consumed, but is being prepared for enzymatic degradation. Now, from biodegradation, we move. The next phase of degradation is biofragmentation. Yeah. So now, basically, there are three steps to the degradation of plastic by microorganisms. First is the biodegradation, second, biofragmentation, and third, assimilation. Now, we have explained about derivation. Now, let's move to biofragmentation. So now, once the microorganism is attached to the surface of the plastics, they produce extracellular enzymes, like I mentioned. Now, the extracellular enzyme produced during biodegradation hydrolyzes the long polymer chains. Now, these enzymes now cleave to carbon-carbon or carbon-carbon bonds or ester bonds within the polymer backbone, reducing very large insoluble polymer molecules into oligomers, dermers, or monomers. Remember that plastics are polymers. Now, the enzymes earlier produced during biodeterioration at the stage of biofragmentation, they are being reduces the polymer that is the plastic, reduces these polymers to monomers. You can see from the Victoria evidence, you can see how complete the polymers are formed there. During biodeterioration, the surface microorganism is beginning to put the abiotic factors penetrate the plastics and then give them like a pore holes where the biofilms are formed. Now, the enzymes now are being attached. You can see how these polymers here are being reduced into monomers here. Sometimes monomers, sometimes diamers, sometimes oligomers. Now, but these stages are smaller molecules which are soluble enough to diffuse towards microbial cells. The biofragmentation stage is considered the rate-limiting stage of plastic biodegradation because the high molecular weight and hydrophobic nature of plastics make enzymatic access difficult. Now, remember that we talked about polyethylene, so I'm going to use polyethylene as an example now. For polyethylene specifically, oxidation of the polymer backbone produces short-chain hydrocarbons are called aldehyde, ketones, and fatty acid-like compounds. Now, after the biofragmentation, we move to the stage of assimilation, which is the uptake and metabolism. The resulting monomer and oligomers are transported across the microbial cell membranes. Inside the cell, these degradation products enter central metabolic pathways, different metabolic pathways such as beta oxidation, glycolysis, the tricarboxylic acid cycle, among other things. Remember that in the definition, Magdalene in 2020 made us understand that the end product of biodegradation is assimilation by microbial cells as carbon source to release carbon dioxide. Now, so there's at this point, we have the carbon obtained from plastic degradation, which is used as biomass production, cell growth, and the microorganism is effectively using the degraded plastic as carbon source. At this point, after biodegradation, during assimilation, this microorganism, the monomers already enter into the microbial cells, and they are confidently already using it as a source of carbon. Now, there's a state that is not visible in this figurative expression, which is mineralization. That's the last stage. This is when, during mineralization, this is when the degradation products are completely oxidized and they're releasing the final product, carbon dioxide and water into the environment. Now, this explanation gives us the process of biodegradation by microorganisms. Now, moving forward. In the last video, I mentioned that in this YouTube channel, we'll be talking about experimental validation, that is the process by which we can experimentally degrade plastics in the lab and in silico analysis. So first, we'll be diving into the experimental process. followed by in silico analysis using computers. So now these are the materials and method when we intend to carry out plastic biodegradation in the laboratory. We know that as scientists, we need to first test these things before we have to test them in the lab and ascertain that the processes can give us results before we finally push it out. So now, for the purpose of this video, we're going to be using this material method. First is isolation and characterization. What do we mean by isolation? How do we go about isolation? Now, we can use different samples because now we're using soil and water. And the reason why we're using soil and water is because when we check around us, plastics are found in either on land or in water. So now, because this environment, this habitat are already contaminated by the plastics, you know what we call adaptation. Thank you. So now, because these plastics are already used to this environment, it will be easy to pick samples from this environment because they already have an interaction with them. So now we could pick either soil samples from an environment that has been polluted with plastics. It's important that we pick them from an environment that has been introduced to plastic because there's already an interaction between the microorganisms in the environment and the plastics. After we pick these samples, we do a serial dilution to a factor of 10. If we are using water, it's easy to do a serial dilution in water. But if we are using soil, it's important that we pick 1 gram of that soil and dilute it first with water. Then we'll further do a serial dilution. After serial dilution, we'll do plating. We'll carry out plating on the agar to identify and isolate the microorganisms that are involved. For instance, if we're trying to do bacterial isolations, after serial dilution, we prepare an agar and do a plate streaking to identify the bacteria. After we streak on the plate, we try to check the morphology because, of course, different bacteria have different morphology. We check for their color, check for their colony, appearance. All of these are pointing to the identification of the bacteria because different bacteria with different colors, different shapes, and different appearance. Then afterwards, we like to know, it's important to know if they're gram positive or gram negative because, of course, the gram state of the microorganism also has a contribution into what they do to us for the environment for that. Then, after doing a gram stain, we carry out a lipase test. Why do we carry out a lipase test? The lipase test is like a gold standard for the purpose of plastic degradation. Now, the lipase test is the gold standard because studies believe that lipase positive bacteria are able to degrade plastic because they have the same ester bond, because they share similar ester bonds, especially polyethylene. Now we're going to use lipase as the gold standard. And for bacteria that were isolated from the soil or water that are positive for lipase, there's a 50% chance that they'll be able to degrade plastic because they have similar polymer structures. Then afterwards, we'll carry out other biochemical tests, which involves proteolytic test, yes, cellulose test, xylanase test, among others, because of course these tests also are useful for the identification of plastics. Then after we carry out isolation and characterization, we are going to do an enzyme activity assay. Why is it important to do an enzyme activity assay? Because enzymatic activity assay is used to evaluate the ability of microbial isolate to produce extracellular enzymes that may contribute to polymer degradation. Amongst these enzymatic assay, acellase activities, xylanase activity, proteolytic activities, and also lipase activities. Now, let's start from the left of this diagram. On the left, the diagram illustrates the mechanism of enzymatic hydrolysis. Microbial lipases and esterases hydrolyze ester bond present in polymers such as polyurethane and polyethylene, polyethylene terephthalate, among others, breaking the long polymer chains into smaller molecules, including terephthalic acid and ethylene glycol, which can be assimilated by microbial cells. Remember when we talk about biodegradation, biofragmentation, and assimilation? Yes, where our assimilation is coming off. Now, to determine whether our isolate produce extracellular enzyme, we have to culture them on selective agar media containing specific substrates. Now, this cellulose activities has cellulose as like CMC as the substrate. Xylanase activity also has xylanase as the substrate, and proteolytic activity has skin milk as the substrate. Now, also, our lipase activity, we also use tributylglycerol as the substrate. Now, what happens if an organism secretes the appropriate enzyme, the enzyme diffuses from the colony into surrounding agar and hydrolyzes the substrate. Now, this degradation process creates a clear zone or an halo. This is an halo. This is an halo or a clear zone around the colony. Now, this colony or halo, they are expressive of enzymatic activity. So we can see that, oh, this is positive. It means that this bacteria has the ability to degrade plastics. Now, so talking about this halo forms because the enzyme is secreted outside the microbial cells and diffuses through the agar medium. As it diffuses, it hydrolyzes the substrate surrounding the colony. After incubation, either the substrate itself disappears or a staining agent. Sometimes we can use the staining agent. Yes, we sometimes use a staining agent to produce this, to make the halo visible. We're going to also, this is like an introduction, this is an explanation of the materials and method of our about degradation about degradation process of plastic degradation. So we're still going to go deep down into the enzymatic assay where we're going to carry out these activities in the lab. Now, after enzymatic, first in our materials, the method is isolation and characterization. Then we move to enzymatic activity assay, and finally we carry out a biodegradation test. What do we mean by biodegradation test? We want to ascertain that the enzymes the bacteria that produce those halo are actually able to degrade these plastics. What do? We pick a pure colony of these enzymes and introduce plastics into them, either polyethylene, terephthalate, polystyrene, any of the plastic that we decide to use. Now, we pick the colony, a pure colony, we add the plastic in minimal salt medium. We're using minimal salt medium because we remember that the purpose of this plastic are to use the microorganism is to use plastic as a source of carbon. So minimal salt medium does not have any form of carbon in the medium. So we're trying to ascertain that these plastics are using these microorganisms are actually using the plastics as a source of carbon. Now, in this setup, we're going to put this in a shaken incubator at 30 degrees for a minimum of 30 days with the plastics. We're going to cut some amount of plastics in the clinical flask. We're going to add some isolate, just one pure colony of the bacteria and fungi into the mixture, and they'll check them for 30 days. We're going to have the positive control, the negative control, and the experimental setup. This video is an overview of the experimental validation of microorganisms that are able to degrade plastics. Just like I mentioned earlier, that this YouTube channel will be focusing on plastic degradation in two parts: one, experimental validation, and two, in silico analysis. Thank you for your attention, and thank you for watching. Don't forget to like and subscribe. Thank you.